Thank you, Tom. Good morning, everyone. So these are my disclosures. So the agent DCB by Boston Scientific is a paclitaxel coated balloon. It has 2 micrograms per square millimeter, and the excipient is basically a citrate ester. It's a crystalline balloon. So they did some preclinical work and showed that this crystalline transpax formulation results in a sustained concentration of paclitaxel in tissue as contrast to amorphous coating. I think this is a very important difference between different paclitaxel coated balloons. I would like to share a patient case. I think this approach we learned from Dr. Colombo. So this is an ostial LAD lesion. We confirmed with IVUS a very tight lesion with small MLA and a big reference size of the LAD and quite healthy or completely healthy left main. So I think the most simple approach is to use a one-to-one sized or a little bit even smaller cutting balloon, do perhaps multiple inflations by rotating the balloon a bit to engage the lesion better. And then after checking the result, non-flow limiting dissections, less than 30% recoil, using imaging if you have on table, to do DCB only angioplasty, in this case with agent balloon 1-to-1 ratio. And this is the final result. There is a very nice MLA, and this patient was asymptomatic during 1 year follow-up. So with this approach, you avoid crossover stenting and pinching of the circ. So agent DCB was compared to sequent please DCB in 1 randomized trial for ISR, 125 patients. And the primary endpoint was 6 months instant leg lumen loss, and the balloons were similarly working in this scenario. And this was not powered for clinical endpoints, but there was no difference in TLR either. So we did a few years ago a registry analysis of all consecutive PCI done with agent, starting from 2014 in our center in de novo lesions. So the patients were approximately 70-year-old. We had 338 patients. Notably, over 50% of the patients had high bleeding risk, 50% or so ACS, and the majority were small vessels, 62%, but we had also a cohort of large vessels, and 72% DCB only, and 27% hybrid approach using also a stent. This is the main result. We followed these patients up to 3 years, but at 1 year, the MACE rate was acceptable given the quite sick population with high bleeding risk and ACS, and TLR rate was 5.6%. And there was also some calcified lesions as suggested by the use of rotablation. This is one case from that registry, quite simple lesion RCA, but there is diffuse disease, and I think by stenting you have to extend the stent length, but by using a DCB approach you can selectively treat the sickest part of the vessel. And again, optimal lesion preparation with a cutting balloon followed by similar size agent balloon. This is the final result, and this is after 2 years, very good result. What we also learned from this registry, which has been suggested also by other trials, is that recoil is the worst enemy of DCB treatment. 74% of the restenosis cases had recoil in angio, if you look back at those angios. Dissections were not associated with restenosis. And if the DCB-only angioplasty with agent was optimized, with no recoil, then the TLR rate was really low, 1.2%. So this registry showed that it was safe and effective in this all-comers population, and recoil was the significant risk factor for restenosis. So then another registry trial with agent came from Italy. This is a little bit larger, 450 patient, 66% of de novo lesions and 71% of large vessels. And it also confirmed real-world efficacy of this balloon with an overall TLR rate of 4% and overall MACE rate of 5.7%. But very recently in TCT, a couple of weeks ago, this Alliance registry was published from Japan, studying a real-world registry of paclitaxel-coated balloons for coronary artery disease. They had also about 70 years old patient, quite a lot of diabetics, 44%, HPR again over presented 56% in this cohort. De novo lesions almost 100%, osteal lesions 21%, quite a lot of calcified lesions, diffuse disease. So this is a quite complex, actually, anatomical population. And which is different from Europe, they used almost 90% of intravascular imaging. I think this is a really, really great approach. And 73% agent balloon in this registry, and 1/4 of sequent please DCB, and very low amount of bailout standing 1.1%. So they had this pre-specified non-inferiority margin of 7.5% for target lesion failure, and this registry met that by 4.7% of target lesion failure, and also the numbers of other ischemic endpoints were low. So this Japanese registry showed that, you know, with intravascular guidance, almost 90% of cases, you can have very good real-world results in a quite complex patient and also anatomical population, and actually met the primary endpoint. So I would like to conclude my presentation that paclitaxel crystal-coated balloons have shown a very good safety profile. For example, in a meta-analysis published by Bruno Scheller a few years ago, paclitaxel, as we have discussed, induced more positive remodeling than limus-based drugs. And for de novo lesions, agent DCB has shown good clinical efficacy in real-world observational studies. And for ISR, we got randomized trials, 2 of them, agent against sequent please in agent ISR trial, and then in the US we have agent against BOBA in the agent IDA trial. Thank you for your attention.